Since her thesis at the University of Orsay and during 2 postdocs at the University of California, San Diego, then at the National Research Institute for Agriculture, Food and Environment in Versailles, she has studied the mechanism responsible for changes in genetic and epigenetic diversity in plants. She as well studied the evolution of their reproductive systems using experimental approaches and mathematical modeling. And since joining the National Museum for Natural History, she has focused her work on understanding changes in plant population and communities in relation to human activities. She's particularly interested in the role of agriculture, climate change, and their interaction between plants and their pollinators. Much of her work is based on citizen science programs, which bring together researcher and citizen to jointly improve knowledge of biodiversity. And today's presentation falls within this research interface between plant-pollinator interactions and global change. So I will now invite her to present her talk entitled Plant Pollinators Interaction in a Changing World.
Thank you very much for the introduction, Flora, and thank you for the invitation. I'm really happy to be here. I understand that you had a short night yesterday, so I'll try to keep the presentation lively. And before going into the detail of my talk, this is a slide that you have already seen because it was on when you entered the room. A warning that I didn't do any of what I will present alone, but together with many colleagues. As you know, science is a collective process, and many of these colleagues are either master students, PhD students, postdocs, and their contribution is really tremendous in terms, of course, of time spent working on project, but also in terms of creativity. And I would have done barely nothing without them. I'm only showing faces now, but I'll give you their names once I present the specific projects they work on. And building this slide, I remember that I attended a similar conference many years ago when I was doing my PhD. And during this conference, one of the guest speakers stated something that may be obvious, but that is worth stating, is that science nowadays mostly progresses because or thanks to young researchers. And at the time, I remember that I was shocked because I was considering myself as only a student with really minor contribution. But this is clearly not true. And I hope you are not as naive as me and that you already realized that. But if not, be aware that your contribution is really critical, and keep that in mind in maybe times of doubt if you have any. So now to the main topic. So the general context for my talk, I guess you know it very well. We are experiencing major changes in biodiversity and its various components, which can be observed in terms, of course, of species extinction, but mostly in terms of changes in population abundance, which I illustrated here with the Living Planet Index. But also, this is related to local population extinctions, which affect the species range. Here, the example is the lion with the historical range in red and the current range in blue. And these changes in biodiversity are also associated with changes in ecological functions. There are many different components to that, and just one illustration is the increase in the frequency of eutrophication in lakes or in marine environment, which is favored, for example, by the loss of top predators. Another element of context which is very well known is that we have identified the main drivers of these changes in biodiversity. I'm not going to list them. I assume you know them very well. But despite this knowledge that biodiversity is changing and we know the main drivers, there are still many open questions, some of which are related to the fact that there are huge biases in which components of biodiversity are the most studied. And to illustrate that, this is the distribution of the number of observations in the Global Biodiversity Information Facility database. And probably you can't read really well, but everything that is in blue-gray represent data for vertebrate animals, and the majority of it is birds. Birds are really cool. I love birds, but they're definitely not the majority of biodiversity. And for example, plants here, represented by maybe, I don't know, 10% of data. They represent 80% of biomass on Earth, and they have really important role in the functioning of the planet. They're at the base of most trophic chains. They regulate climate and so on. And insects here, even fewer data, they represent 80% of species. So I don't want to claim that we are only plants and insects are interesting, but they really need to be studied more. And also there are understudied ecological functions. For example, you have a lot of studies on primary productivity because it's easy to collect data using remote sensing approaches. But other functions, and particularly the functions that link plants and insects, are understudied. And second, even though the main drivers have been well identified, we are not sure what are the relative contributions of these different drivers, whether they interact. We know they interact, but there are still a lot of open questions. And we don't know very well what are the roles of interspecific interactions in shaping the response of biodiversity to these drivers. So the question I will address today are to try to describe what the recent temporal dynamics of insects and plants are currently. And because this is the functional ecology session, I will mostly try to examine whether these temporal dynamics impact one major ecological function, which is pollination. Okay? I'll start with a reminder. No offense meant. I'm sure you all know what pollination is, but I want to make sure we're on the same page. So pollination is the transfer of pollen, which is the life form in plants that produces the male sexual gametes. And it's a transfer of pollen between the male part of flowers, stamens, and the female part, pistils. And this is really essential for the sexual reproduction of plants because pollen cannot move by itself. So it has to be transported by a vector. This vector can be wind, but for the vast majority of plants, flowering plants, over 80%, this transport involves an animal vector, which is called a pollinator. And in a large fraction of cases, these animals are insects, especially in Europe, but in other parts of the world, they can be birds, bats, and other types of species. I'll focus on insects for today. And about insects, there has been quite a lot of debate in the scientific community and outside on whether these insects are currently decreasing. With many publications and journalist media over the past decade or so. Here you have an example with the New York Times Magazine. But you had other publications in French newspapers, for example, Le Monde, or in The Guardian. And you can see that the vocabulary used here is quite distressing, with words such as apocalypse or catastrophe. And these articles have been prompted by a number of scientific publications alerting on a possible global decline of insects, and with a warning that was issued by the French Academy of Sciences, for example, in 2021. So there seems to be a consensus on this decline of insects, yet there is still some ongoing controversy on whether insects are declining or not. And to illustrate that, you have here 3 scientific publications that appeared roughly at the same time in different regions and on different groups. The first one at the global scales was a meta-analysis of time series that demonstrated decline in terrestrial invertebrates or insects, although the paper did not include insect only, but increases in freshwater insects. The second one on the United States showed no net insect abundance declines. And the last one in Great Britain, focusing on invertebrates, including insects, concluded on complex changes. So there are really different information carried by this different data set and analysis. And with several colleagues, including François Duchesne, who was a PhD student then and who is about to be a CNRS researcher starting this year, we we put together the data sets from these different studies, and we tested whether the starting dates for the time series that were used in these papers had an influence on the inferred temporal trends in abundance. This should not matter at what time you start the observation if the change in abundance is linear. If you have a linear trend, you start here, here, or here, you will infer in all cases a decline for this specific example. However, if the temporal changes are nonlinear and even worse, non-monotonic, then it really matters when the observations start. If you start here, you will infer a linear decline. If you start here, you will infer a linear increase. And if you start later, maybe a stronger linear decrease. And what we showed was that if you include the temporal baseline, the starting date of the different time series in the analysis, then you have no differences in conclusion between these different studies. Another important conclusion is that it's really complicated to conclude, to give any valid conclusion on the temporal dynamics of insects or any other groups with data sets that are highly heterogeneous and highly biased. And in general, there is a lot of interest for the big data approach. You should keep in mind that big data have limitations, and in particular, they often are associated with a lot of biases. And just to illustrate that, I'm using an example that was published by colleagues from the UK with a plant species, heather, which is very convenient because heather forms large patches of purple to pink flowers, which can be detected with remote sensing data. So we can know the true distribution of the species with such data, and it's mapped here in Great Britain. All the green parts are the distributions of heather. Great Britain is also a fantastic country to analyze ecological data because there are a lot of naturalists who are really eager to go in the field and collect data. And here you have the map of the sites, which are squares of 1 kilometer or by 1 kilometer, that have been visited at least once by a botanist. There are almost 20,000 of such sites, which is huge. In France, we barely managed to reach 200, maybe 2,000 sites, but 20,000, no way. But you can see that although there are a large number of sites, for this species in particular, the distribution of observers does not really match the distribution of the species. And in fact, if you look at the sites in which the species was observed, there are really few. And what these colleagues have shown is that with such a biased distribution of observation, the abundance of the species is estimated as poorly as with 28 sites that would be randomly distributed and non-biased across the country. So big data is cool, but they're biased and they cannot always be corrected for. And with these colleagues, we emphasize that it's really important to, at the minimum, report the biases that exist in your data. And we provide a tool to do so. And when possible, correct for these biases. And to think twice and be inspired by these two persons who published a paper to discuss the use of big data, and that is entitled "Big Data is the Answer." But what is the question? It really depends on the question you ask, and sometimes big data, the number of data does not correct for their biases. And just to finish with that, I would like to emphasize also the importance of structured data. Structured is meant in opposition to opportunistic data that are collected without any specific protocol, particularly to infer temporal dynamics. So structured data, I mean data that are collected with systematic protocols and using a well-thought sampling design, which makes them reproducible and comparable across sites and through time, and also representative of the diversity of the area you're studying. And for that, citizen sciences can be really interesting tool to collect such data. And in fact, for the case of insects, the only reliable data that did demonstrate a decrease in the abundance of insects or data that were produced by non-professional, non-scientist persons. Either you may have heard about this initiative in the UK, Bug Matters, in which they offer citizens to clean the mineral plate on their car, drive, and then count the number of insects that were dead on the plate. And with such approach, they demonstrated a 63 decrease in the abundance of insects over the past 4 years. And you may have heard also, this is an older publication of this work in Germany, trapping insect and just weighing the biomass that was trapped, showing the same order of magnitude, 75% decrease in biomass over the past 30 years. These are purely citizen science data. And I'm just advertising. In France, we have many citizen science programs, but one of them is led at the Museum of Natural History. It's called Viginature. And several of the observatories within Viginature focus on insects. And I have been working with the Farmland Biodiversity Observatory, which is directed towards farmers who can monitor wild species in their fields and also inform on the farming practices. And this was the work of Olivier Billot, doing a PhD. And he showed that with such standardized protocols, simplified data collection, farmers do not identify species, it's not possible, for wild bees and butterflies, he documented a decrease in abundance through time that was related to farming practices, and the decrease was, as you would expect, stronger where farming practices were more intensive in terms of pesticide use and fertilizer use. Okay, so this is the functional ecology session, so let's go to changes in function. So I already said that animal pollination was an important ecological function because it controls a large fraction of wild plant reproduction, but also it is important for crop yield for many crop species. And we know very well how to estimate the effectiveness of animal pollination. But this requires experimental determination that is really time and resources consuming. This requires either supplementing pollen on each flower individually or bagging each flower to exclude pollinator and look at how seed production is affected, etc. So it's virtually impossible to measure pollination over large areas or over long periods of time. There are a couple of examples that did it, but it's really rare. So what is done generally is to use indirect measures of pollination when we need to estimate animal pollination over large areas. So for example, when the data exists, we can look at the abundance or diversity of pollinator communities. And another widely used index is even more indirect. It uses land cover and climate data to estimate where resources will be available for pollinators in terms of food resources and nesting resources, and to map potential presence of pollinators. So it's really completely indirect. And with several colleagues, what we did is to try to provide a direct, realized measurement of animal pollination. This was the work of Gabrielle Martin during her PhD. Gabrielle is now assistant professor at the University of Toulouse. And with the help in the initial steps of master student Martha Macro, this realized pollination index uses data on crop yield. This is available in France through the Ministry of Agriculture with a core spatial resolution, which is the administrative department. And at the time, we had data over 2000, 2010 for 55 crops whose yield depends on the production of seeds or fruits, so depends on pollination. And we also use data on how much the yield of this crop depend on animal pollination. And this is an index that varies between 0 and 100%, and that measures how much yield is reduced when pollinators are excluded. So this goes from no effect of pollinator exclusion for crop independent of pollinators, such as bread wheat or other cereals, to crops that are completely dependent on pollinators. If you remove pollinators, you have barely any production of seeds or fruits. And to calculate the index, the first step is a standardization step because we are literally comparing apples and oranges. So here, for example, if you look at the yields of different crops such as, for example, oilseed rape and cucumbers. Cucumbers, they have a lot of water, so the yield is really large. And we want to be able to compare. So we standardize and we have the same distribution across the study area, which in the first example would be France. And the rationale for obtaining the index is simple, but then there are technical details behind that I will not explain because I don't have time, but we can discuss later if you want. The idea is all else being equal, and that's very important. In an area with pollination deficits, we expect the yield of pollinator-dependent crops to be lower on average than the yield of pollinator-independent crops. That's because pollinators are not sufficient and the yield is affected, but only for pollinator-dependent crops. So for example, in an area with such pollination deficits, if you look at the relationship between these standardized yields and the dependence of crops on pollinators, then you expect a negative relationship because pollinator-dependent crops suffer from pollen limitation. In an area where you have no such pollination deficits, you expect something flatter. And the index of realized pollination is simply the slope of this relationship. And we applied that to France as a whole. And this is what we get. Here, the pollination index is higher with orange colors and lower in blue colors. And you have a strong latitudinal gradient, which is quite consistent with expectation because in France, intensive agriculture is here. You also have a climate that is less favorable for pollinators, and you have fewer semi-natural elements in northern versus southern France. And also, remember I showed you this independent measurement of pollination on the basis of land use and climate that had also this latitudinal gradient for France. And if you look at the correlation between the realized pollination index and this potential pollination index, we have a strong correlation between the two. Even though the data used to calculate the two are completely unrelated, crop yield here, land use here. So we are confident that our index does measure animal pollination, but the relationship is not a perfect one. So we hope that it carries more information. And we went a step further because the scale of France was not very convenient to go into the mechanism responsible for this variation in pollination effectiveness, because you have a gradient in climate, in intensity of agriculture, and in semi-natural element that are related with each other, so it's hard to tell apart the different roles. So we did the same approach, but this time at the scale of just a landscape, using an experimental site of the Institute for Agronomic Research, which is a site near Dijon in France. So it's about 1,000 hectares. And this time the spatial resolution of the data is the single field. And we have a range of years for data, and of course, a smaller number of crops because the area is smaller. And I won't go into the details because I don't have time, but we calculated the index at different spatial resolution from really the single field to groups of fields, and I don't present the whole details. But what we get is the same. On top, you have the potential pollination that is predicted on the basis of land cover and how much resources are present for pollinators and potential pollinator presence. And on the bottom, the realized pollination index based only on crop yield data and pollinator dependence of these crops. And I find it really amazing that the two maps are quite similar, although there are differences. And these differences might be explained in part by factors that are not included in the potential pollination index, and in particular, farming practices. And what we show that the covariates of the realized pollination index is in particular farming practices when the index is calculated with a fine resolution. And in particular, we observed a strong negative relationship with pesticide use. Even though you have a lot of resources for pollinator at a given place, if farmers use a lot of pesticide, the realized pollination is lower. And at coarser spatial resolution, when you pool several fields together that can have different farming practices, then the main driver of the measured realized pollination is landscape elements and the presence of semi-natural elements. An interesting thing with the index is that it can be calculated in time as well. So instead of separating the study area into different sites, we can separate in different years and look for different years whether you have a negative relationship between yield and pollinator dependence or not. When we do that with a study at the landscape scale, we didn't get any significant linear trend, but there is strong year-to-year variation in pollination effectiveness that is related to weather condition. And this has still to be examined into more detail. And at the national scale, this was the work of Lena Brugger. When we analyzed the dataset without asking too many questions, this is what we got. An apparent linear decline in the effectiveness of pollination through time. But then we looked at the data in more details, and what we saw is that the Ministry of Agriculture changed the method to report yield through time in 2000. So really, the two different time periods cannot be compared. And when you separate the two time periods, you have no significant linear trends. So I haven't updated this for 2025. I will eventually. But this is a reminder that really we need standardized long-term monitoring. And if you change the method in the middle of your time series, it's really a nightmare. Okay, so here, these analyses suggest that even though we don't have a temporal pattern as of now, that there are deficits in pollination, at least for crops, but we can wonder what happens for wild plant species. What happens for crops probably has impact also for wild plant species, but using data on wild plants could be another angle to detect possible changes in the effectiveness of animal pollination. And this, we can do it using another citizen science monitoring scheme, which is called Vigiflor. It's a scheme that involves highly skilled botanists to monitor plant communities across France. Here you have a map of the monitor sites since 2009. The idea is to have a random sampling of the territory, so you can see that we did not really achieve that. Standardized monitoring also have biases. They're probably a bit less than opportunistic data, but you still have biases, and they need to be accounted for in the analysis. But still, we have a reasonably good coverage of the territory. And the data collection is highly standardized. Botanists had to count the presence of species in 8 systematically distributed sites within a square of 1 kilometer by 1 kilometer. And each of these sites is made of 1 square meter plots in which they note the presence or absence of all flowering plant species. So this makes comparable data, and also it produces data on abundance, which is abundance of species, which is more sensitive to temporal change than the just presence absence. And the scheme started in 2009, so we now have over 15 years of data, and it is possible to compute temporal trends in species abundance. So here you cannot read anything, obviously, but it's just to show that we have temporal trend for over 600 wild plant species. And you can see that some species are increasing, others are decreasing, some are stable. And you can try to explain what causes this variation in temporal trends. And there are many, many different causes, but for the purpose of this talk, I will focus on pollinator dependence, which is one obvious reason that may explain differences in in temporal trends. And this is the current work of Solène Agnew, who is finishing her PhD, and also with initial contribution of Etienne Henry, who is one of the co-organizers of this conference. Quite a few years ago, I think it was during COVID, right? And one expectation we have is that we have a lower average temporal trends for plants that are highly dependent on pollinators for their reproduction versus plants that are not dependent. And this is sort of what Solène observes. Here you have, for all the 600 plant species, the temporal trends in abundance for mostly wind-pollinated species, mostly insect-pollinated species, and others. It's not obvious, you'll have to trust me, but the difference is significant with this approach. The temporal trend of insect-pollinated species is lower than that of wind-pollinated species. The one thing we don't understand very well is why the other group is even lower than the insect-pollinated plant species. And another phenomenon that Solène observed is that this difference in temporal trends between wind and insect-pollinated species has consequences for the composition of plant communities. And there is a strong tendency for loss of pollinator-dependent species, which is particularly strong in cropland, but which is true also when pulling all habitat types together. Here on this graph, you have the average species richness in one 10 square meter plot as a function of time. Here in, this is the graph for croplands. And by looking at wind-pollinated plant species, number is stable, and insect-pollinated species, and the number of species is decreasing linearly. And we tried to examine whether this pattern that we observed in France is valid more broadly outside of France. And this was the work of Lucien Desplanches, who was doing an internship, a master internship, and with the help of Clément Vallée and also Solène Agnew, and of course, all the partners of the different countries involved. Lucien tried to synthesize the same type of approach. Is there a difference in temporal trends between insect-pollinated versus wind-pollinated plant species for 9 plant monitoring schemes that went from scales either regional. Here you have data from the different botanical conservatories, which are in France the entities that are in charge of monitoring and conserving plants. And national schemes in France, in the UK, in Denmark, in Switzerland, and a regional scheme in Spain. And the purple ellipses here is the forest replot scheme in which you have permanent plots that are resurveyed over large time periods throughout Europe, mostly Northern Europe, and also a little bit in North America. And this is a summary of what Lucien observed. The general pattern is similar. That is a tendency for temporal trends in plant abundance that are more negative with pollinator-dependent species than wind-pollinated species, but the signal is not very strong. You can see that we have only, so here when you have negative value, it means that pollinator-dependent species have more negative temporal trends than wind-pollinated species. So in 4 countries, you have this pattern. You may notice that for France, Lucien used a slightly different type of analysis, and he didn't find the same result as Solène. So we'll still have to solve the issue here. But you have even a case in the UK in which insect-pollinated species have more favorable temporal trends than wind-pollinated species. So this is still ongoing analysis, so I don't want to conclude very strongly on this, but there still is a tendency for more negative temporal trends for insect-pollinated species, and the differences can be explained by the starting date of the different observatory. As I told you, standardized monitoring does not mean that you don't have any biases. And there are differences in starting dates. There are differences in which species are monitored. Do we have data for rare species or for common species? For example, in Vigiflore, we can only calculate temporal trends for species that are abundant enough that we have data to run the statistical analysis. Whereas the botanical conservatories data, they have a lot of observation included for rare species, and they can compute trends also for species that are more rare than what we do in Vigiflore. So we have to still dig into that. Okay, but I'm reaching the end of my time, and I will conclude. So I think we have now sufficient pieces of evidence that plant-pollinator interactions are changing with functional consequences in terms of animal pollination, which seems to be deficient in at least several areas of France and Europe, and probably globally. However, there are many other drivers at play, and I, with colleagues, are working on some of these other drivers, which are climate change, impact of land use, and particularly agriculture. Yeah, that's the main two drivers I'm considering. And we still need to go into the details and try to disentangle the different contribution of these drivers and how they interact with each other. And I'll finish with some advertisements on a possible funding opportunity. We are in the process of asking funding for a new PEPR. The French among you may know what this is. This is a new tool to fund research. And I co-lead with Rodolphe Rougerie and Wilfried Thuillier one that will be called Dynamique de la biodiversité terrestre. If it is funded, which is likely, but we're not completely sure yet, there will be 40 to 50 million euros dedicated to studying the dynamics of terrestrial biodiversity. So stay tuned for upcoming opportunities. And thank you very much for your attention. I'd be happy to take questions.
Hello, thank you very much. I was wondering, so most of the studies you showed, I think they started not earlier than the 1990, so it seems quite hard to have like earlier data on it. Are you aware of any approach using maybe indigenous knowledge, or like asking people if they think there were more insect before, or maybe if the pollination was better? Are you aware of such approach?
Yeah, definitely all the time series I'm using, and even generally all time series started really late in comparison to when the major impact of humans on biodiversity started. And this is one of the aims of this research program, is to try to go back in time earlier. So a lot of it will be focusing on collection, physical collection. So trying to infer time series with these highly biased also data sets. But there are other options, and I know some in which is called historical ecology, going into different sources of literature to try to find some information on the presence and ideally abundance of species. Definitely indigenous knowledge may be part of this general approach. But all these approaches for collections and historical ecology, they're still in the beginning. And the idea is to expand them with such a program.
Thank you very much. It was really great and inspiring talk. I was wondering, so you showed like a decline of insect-pollinated species, and I was wondering whether you said it's even more in cropland, and I was wondering whether there would be habitat where they are doing better. I'm thinking of edges and things like this, and like the variation between habitats of these trends.
Yeah, I didn't show all the results, but there are for Solen's analysis on the temporal trends of wind pollinated versus insect pollinated species. The general pattern is that the species richness of insect pollinated species declines, but there are differences across habitats. And for example, in grassland, she observes a tendency for an increase in the species richness of insect pollinated species. Which we don't explain very well, but there might be changes in practices, for example, that favor the return of insects and insect-pollinated species in these habitats.
When you showed the change in plants abundance collected with the Behaviour program, just briefly these graphs, we could see that there are many non-linear patterns. They seem to be non-linear. I can see, of course, the value of approximating it with a linear trend, but do you see any way of maybe accounting for that or taking it into account somehow?
Yeah, definitely it's possible. So far we have been doing the straightforward basic stuff, just looking at the linear trends. But we do need to go into details, and I know there has been some work here at Fith to report, to provide tools to describe all the different nonlinear patterns in the time series and to report them and explain them. So yeah, this is definitely a perspective.
Was there a question?
Thanks for the talk. In the first part, where you showed that the time scale depends, it's very time scale dependent whether you find a decline or not for the insects. But doesn't this argue that there's no trend, in fact? If it depends on the time scale, it means, and you showed it on the graph, it's fluctuating or, so anything that is so much time scale dependent is sort of an argument for there not being clear trend, more like fluctuations.
You could still have a trend.
Yeah.
Like in this example here, the idea was to illustrate a situation with a trend, although it's not obvious, but in which you can still infer different linear trends depending on the starting date.
Like some noise over the trend and things like this, or fluctuations beyond the trend, something like that.
Yeah.
Different embedded timescales.
But the general conclusion of this paper is, as of now, given the statistical tool that we have to correct for bias, it's really dangerous to drive any conclusion because the data are so heterogeneous and so biased that yeah, we cannot say much with that.
Okay, thank you.
Okay, thank you.
Oh. If you have more questions, we can discuss at coffee break, because I'm really sorry, but I have to leave before the end of the session this morning. I wanted to stay the whole day, and I planned to stay the whole day, but one of my trains was cancelled due to the heat wave, so I have to leave earlier than I thought. It's really impolite, and I'm so sorry about it, but I will be available at coffee break.
Thank you very much. Thank you very much, Emmanuel, for joining us at the conference. It was really nice seeing you today. So we are going to continue with the oral presentation from the participant of the functional ecology session. And we now welcome Barbara Bignon, affiliated with the National Museum of Natural History and Paris-Saclay University. And she's going to present us the prototype shape of burrowing and non-burrowing cockroaches moving in granular media.
Okay, good morning. So I'm Barbara. I'm working at the Museum d'Histoire Naturelle and the Université Paris-Saclay. I'm happy to be here today and that you are here this morning. So I'm gonna present part of my thesis work, which focuses on insect burrowing on granular media. Okay, donc as you may know, cockroaches are known for being common pests, but they actually show a wide variety of shapes and behavior, and many of which are still unknown. Some cockroaches are capable of burrowing into substrate like sand or soil, and we might wonder whether some of their shapes are particularly suited for burrowing into their substrate, and possibly influenced by the physical forces in their environment over time. To understand how body shapes evolve, we need to study how insects burrow, but what kind of environment they live in. This helps us identify the forces at play and understand how the insects move through it. And soil and sand, where some cockroaches are known to burrow, are granular media. And granular media also include snow, Pale's green, and even satyr's wing. They are heterogeneous material made up of grain larger than 100 micrometer. At large scales, grains behavior depends on collective motions. Most studies focus only on simple shapes like sphere or cylinder. Measuring drag, it's the horizontal resistance, and lift, it's the force that can push objects to the surface. In this project, we study how these forces behave with more complex shape, especially the pronotum of cockroaches. And what is the pronotum? It's this. It's the first thoracic segment, but it's the first part to interact with the substrate during burrowing. Oh, I'm sorry. We want to know if shape influence the forces during movement. In other words, which shapes face least resistance. So my project combine ecology and physics. I promise you there is no equations. We wanted to know if shape sorry. We study how shapes behave in granular media from a mechanical point of view, and the goal is to connect shapes with physical forces to understand the mechanical rules in this environment. So to do this, I printed 8 pronotum from cockroaches, and I tested them in 2 regular test rings filled with glass bean, one with 1,000 micrometer grains and the other with 200 micrometer grains. And we have a mechanical arm with a 3-axis sensor, move them forward into the substrate. With this setup, I conduct 2 experiments, changing 2 parameters, the depth of burial and the angle of intrusion. So first, I buried the pronotum at different depths, just under the surface, 10, 20, and 30 millimeters under the surface. This allowed us to observe how these forces change with burial. In the other, I vary the entry of my pronotum into the substrate. We wanted to see the effects of angle on the measured force, and if there is an optimal angle that minimizes drag and lift, making burrowing easier. Each test was repeated 3 times, and the 3-axis sensor measured drag, lift, and force and lateral forces. It's the only question. Donc, using this standard equation, I calculated drag and lift coefficients. Sorry. Which reflect how streamlined the object is. So lower coefficients mean less resistant, so more efficient to move into the substrate. Here we have the force, it's the measured force, the exposed surface of the model, the effective density is the rho on the substrate, the gravity, and the depth. So here's the result for the drag coefficient for the first experiment. We have all spaces with 3 spaces known to burrow, and a point on comparison with a sphere. We can already see that the drag coefficient is higher in the smaller grain. For some species, it's almost twice as hard to move through it. And we can see that a little. Species exposed to higher drag are not soil and sand burrower. We have the same result for lift coefficient. Here we can see the same trend. They are higher in smaller grain for many species. And we can see that burrowers show lower coefficients, even negative, meaning they tend to stay buried into the substrate. So for the angle, donc we can see a clear minimum at 1.8 degree, where the drag and lift force I are minimized. So it can be an optimal angle to enter into the substrate and burrowing. Beyond this angle, the forces increases again. Okay, so until now I've shown the forces measurement as graphed, and next we wanted to know the link between the forces and the pronotum shape. So I use a 3D geometric approach to compare statistically the shape and the forces. So first, I place landmark. I don't know if you know it, but on all my 3D models, and I use R software to adjust them. I run a generalized progress analysis. It consists in translating, scaling, and rotating all the model to focus only on the shape. And then I run a phylogenetic partial last square analysis, which tests how 2 datasets vary together. In our case, it checks whether changes in pronotum are associated with changes in the drag or lift coefficient, considering phylogenetic relationships. So in the resulting graph, we will see later, the x-axis shows the shape scores, so it's simplified value that represent the difference in pronotum across species. And the other axis, it's forces scores that summarize how drag and or lift vary. So this is not the original value that we will see, but new value calculated by analysis to capture the link between shape and forces. Okay, so this is the graph, the resulting graph for drag coefficient for smaller grains. We can see the shape of the pronotum. In gray is the minimum shapes, so it looks like the spaces on the right, on the left. And the result, the orange shape, is the maximum, so it represents the shape of the species in the other. Here we can see species share similar morphology and similar drag coefficient, and on the left and on the right, we can see species who have very different shape and coefficient. Okay, so just up. Okay. We have a significant and strong correlation for the smaller grains for drag coefficient. We have species who burrowing tend to group together, but with two other species non-burrowing, and Panestia, who is wind burrowing, so she digs galleries into deadwood, not into granular media. For lift coefficient, we also have a strong coefficient for the larger grain, but not for the smaller grain. But this is a graph for the smaller grain. We can see that burrowing species share similar morphology and similar lift and drag coefficient, and differ from non-burrowing species. So to summarize, the coefficient for lift and drag are higher in the smaller grain. We have an optimal angle at 1.8 degree that minimize the forces during burrowing. And we found a correlation between pronotum shape and forces, meaning suggesting a link between morphology and burrowing efficiency. So, so this helps us better understand how granular media behave with more complete shape and how biological systems adapt to this environment. And next, I will add more species to improve the robustness of the dataset, because it's just 8 species, and the analysis. Thank you for your attention.
Hi, thanks. I was wondering whether you could compare your results with the like a biological model and then just look at the species burrowing into the like a defined soil, and whether you could like you would give you like clue and interpretation of like compare like the results you have with the experimental work with the actual biological data, and whether it's an option or
Yes, with the other insects, for example, or other,
no,
other
like
things
looking
that
at.
burrow.
I'm sorry.
Other things that burrow like microcoaches.
Yeah, like looking at the insects going into the earth and you know with gallery and glasses so that you can see them going through and like compare it like your ideal angle and whether the insects go at that angle or not, things like this. Just
Yes, exactly. There is a big lack on this study. This is very understudied. So locomotion on the granular media are studied, but not under. So there is no very study who I can compare my results. It's very preliminary results for this. For the angle, in the other part of my thesis, I will look how living species burrow. So I can look if it's an angle of 1.8 degrees, but not in other studies. Thank you.
Yeah, thank you for the talk. I was wondering these different granula, they might change in their structure when there is, for example, rain, or if it's actually very dry. So how do you think this environmental change might actually play a role in shaping also the morphology of the cockroaches?
Yeah, the two granular media. What, can you repeat, please?
Yes, what I mean is like these granula. So if they are, for example, in the sand or in some other type of granula, if it's like if all of the sudden it's like very wet, so how the sand can compact, for example, how does it can also drive maybe basically the shape that you are now explaining.
Yeah, yes. For the sand is very wet. It's wet, but the soil, some species burrow into dry soil. In the experiment, we want to add oil that represent the water. Okay.
Thanks a lot. I was, no, I had a question. I was wondering, when you study the individuals and how they bury, will you also be interested in the intra-specific variation? Because so far you have been focusing on intra-specific variation.
For now, we're just focusing on yes, in interspecific, yes, but because we have not much species, it's just a species.
Okay, thank you. Now you can go.
Thank you, Barbara, for your presentation. And we are now welcoming the next participant. We are welcoming Chloé Vagnon, affiliated with Center for Synthesis and Analysis on Biodiversity and Functioning of the Vertebrate Scavenger Guild.
I want to find out if I'm louder than this mic. I don't think I'm that loud. Anyway, I'll be talking to you today on the role of large carcasses on the structure and function of the Vertebrate Scavenger Guild. And I did the study in southern region of Africa. So in this place, scavengers are like under threat. Most of them are critically endangered, but we know so little of them. What's interesting is that recent press shows how many scavengers we are losing every day. Like this publication is from last month. So, but then it is because of poaching in the region, right? Because elephant carcasses, for example, they contain tusks which poachers actually are interested in. And somehow the non-target species, like the vultures, they don't know if a carcass is poisoned or not. And while they're feeding on them, they might get poisoned, and a lot of them die in high numbers. And then for lions and other mammal species, a lot of poaching happens in the region. But then again, these species are under threat, and we know so little of them in this area. But what we know is that they have a high economic value. For example, this recent publication showed that the scavengers, they contribute to like 250 million US dollars per year, which is a lot. We also know that, like this BBC publication, which I cannot explain, they say that vultures contribute a lot and their absence might have contributed to that. But anyway, elephants in general, especially in this region, we know that they contribute to the ecosystem in terms of shaping the ecosystem, but as well, they also die due to climate-related causes naturally without any poachers. We also know that scavengers play a big role in like breaking down the molecules into the soil, which is very good. And they also help us prevent the spread of diseases. Good, right? So where do large carcasses come in? If you look at a small carcass, for example, most species would feed on it within like an hour or 30 minutes, and it's difficult to monitor them. But if you have a large elephant carcass, it can stay in the field for a long time, and you can monitor what's happening to them. So in this area which I studied, there's some research on lions and hyenas, but then there's not much research on which scavengers are there in the region besides lions and hyenas, because we know them as hunters, but we don't know them as scavengers. So there's also some research on scavengers in like the entire world. So I use some of these models to model what is happening in this region. So my first objective was to find out which species are feeding on these carcasses, who are they feeding with, or what time are they feeding, right? So for my presentation, I'll be looking at, I'll be calling large carcasses, these are adults, and the juveniles, these are small. So small, it's a small elephant, baby elephant, but it's still big relative to other species. Large, adult elephants. My second objective was to find out while these species are feeding, what are the probabilities that a species might feed, and also how long do they feed? So, which is my second objective. So I did this study in Zimbabwe, Hwange National Park, which is in the southern region of Africa. What we know about this place, there's a lot of elephants in that area, and these elephants are still affected by the climate-related causes that I said. And there's a lot of publications about that place. And also that research that I told you about, the lions and hyenas. So I put up camera traps in 2022 and 2023 on elephant carcasses that I found randomly. I did not poach or kill any of those elephants. They were just there naturally. So I found 11 scavenger species, which is interesting. None of these scavengers were actually known to exist in this place. What was interesting to me was how they fed on these carcasses. Blue represents small, the juvenile, the baby ones, and red on top there represents the adult elephant carcasses. So at the bottom, you see many scavenger species and how they're spread. Trust me, this is not a random like distribution. So if you look at nestedness on its own, it is a group of species that perform a certain behavior that is linked to other species around. So on your far left, you see generalists. These are species that also hunt and scavenge at the same time. I'm not sure if you have enough time to look at the numbers and which species are there. But on the right, you find that these are more specialist species. That means they feed mostly on the carcasses, right? They depend mostly on carcasses and they cannot hunt. If you look at the blue and the red lines, these are the first stages of decomposition. This is how I spread them. My next slide will show you which stages are there. But the darker lines represent like the later stages of decomposition, when it's like maybe after like 3 weeks when the carcass is dry and there's not much stuff to feed on. That is me on the far left. When the carcass, before I set up camera traps, and on the far right, this is when I like monitored after some time, and the carcass was very much like decomposed. The bottom images that are there, this is camera trap information. On the left side again, these are species that are feeding when the carcass is still a bit fresh, not really much open. And on the other side, it's like after some days. This is when specialists only are coming to feed. And the ones that feed when the carcass is still fresh, these are mostly hunters, they're not there. I also found that there's a big overlap, like a relationship between how these species feed. The ones that have a dark blue color are the ones that are mostly feeding together. You don't have to look at exactly which species are feeding together. But anyway, what's interesting, you find that the bird species, the vultures, they mostly fed together and they could cooperate while they're feeding. Whereas the mammals in the brighter colors, they were like avoiding each other. So mostly because of conflict or they would fight. So I went ahead, I wanted to find out what exactly is happening in these carcasses between the large and the small ones. On the large ones, you find that most of them, of the species, these adults, you find some species were coming in abundance. You get a lot of them feeding at the same time. Whereas in the smaller carcasses, the baby ones, you find fewer species in terms of numbers. But what is interesting is that if you look at the timelines between 0 and 6, this is midnight, and also the far end between 21 and 24, it's like 2400 hours. You find mostly the mammal species feeding, as you can see on the images there. Whereas during the day, you get a lot of bird species coming to feed. What was happening with the stages of decomposition? So from stage 1, when the carcass is still fresh, you found that they're also coming in high numbers. But as time went on, stage 4, going to stage 4, you find fewer species, like fewer, like in terms of abundance, fewer individuals are coming to feed, right? Which is also related to that nestedness. Again, so now it's more clear. Also, in terms of activity, when the carcass is still fresh, you get a lot of species coming to feed. But when it gets drier, like on the later stage, you find less activities. This is like on a 24-hour time scale. But okay, we have that information. We know who's feeding, with who, what time, but then how are they feeding? This is where it comes in, like the co-occurrence interactions between these species. It gets more more statistical. So first, I wanted to find out the probability that a species might be present given any 10-minute interval. Do this for the large and small carcasses as well. Like in terms of if it's a large carcass or a small carcass, what is the probability that a species might come to feed? And also, if there's a certain species feeding on the carcass, is the species going to be able to feed? I also did this for the probability to feed, because you might be present but not feed, because you can show up on the camera trap detected, but you may not feed because someone is there. Then I also did this on the proportion of feeding, so that you find while they were feeding, how long did they feed on the large or small carcass? Also, uh, also, I wanted to find out like how long they feed. But anyway, we got a lot of results. But what was interesting is that when you focus on a few individual species, you see a lot of distributions. But then what's interesting is that uh, across all species, they were all able like to be present at any given time. But when you look at the proportion of feeding, right, this is where it gets more interesting. If you focus on the white-backed vulture, for example, you notice that while it was feeding with other bird species, it fed a lot, but when the lion or the spotted hyena, the mammals were present, it did not feed quite a lot. The lion, which is interesting because they always like chase away other species, you realize that it was mostly feeding the whole time. It was mostly feeding the whole time, so as to like chase away other species. So in conclusion, we notice that large elephant carcasses attract a broad range of scavenger species due to their greater resource availability. That allows a more complex feeding interactions with elements of reduced competition. We observed a highly classified assemblage in large carcasses in terms of nestedness, donor overlap, whereas in small random carcasses, the feeding patterns were more random and were less predictable in terms of nestedness. And small elephant carcasses provide an alternative feeding niche for other species that might struggle to access large carcasses with intense competition. And that's all I did. Thank you.
Thanks a lot for the talk. In your study of how the probability of feeding is influenced by other species, did you consider the timing of arrival? Do you have the information to see whether the species arrived before or after another species, and that may help in interpreting whether you have competition or attraction or something else?
Yes, actually, I did consider the timing, because in the sense that if you're calculating the probability of a species to be present at any given time, yeah, okay, on this graph, for example, on the first line, I'm sorry I was very fast because it's a lot of stuff. When it's feeding alone, right, that's when I noticed like this. I'll create a control if you're doing experimental studies when a species first comes and there's no other species there. So that's when it's feeding alone. But any given chance that when it came to feed, there was another like species feeding before, that's when we actually computed that, like, what is the probability of it being present or for it to feed at that given time, given that there's another species present at the carcasses. So yes, I did factor in that. Yeah.
Thanks for the very nice talk. I was wondering how hard it is to find these carcasses, and how do you find them actually?
It's a very good question. That actually led me to my PhD study, which I'm like studying now, where we'll be putting like GPS collars on these animals as they look for the carcasses, which is easier to follow when they're actually stationed at a place for a long time. But when I was doing this for the first 2 years, I was driving in the bush, trying to look for vultures like diving in, or just also the smell of the carcasses is quite intense, so it's easier to find. I mean, these are huge carcasses. But anyway, it was random. Just drive around every day, try to find carcasses, and it's a huge national park, so it wasn't easy. That's why I found very few carcasses as well. Oops, sorry. So those are the ones that I found. So those are the connecting roads as well. So I'll drive every time, try to find anything. You can see that there might be a bias towards closer to the roads as well. It's not my fault. I just wanted to find out who is there. So at any given chance, if there was any carcass in the bush, whatever I found is the same species coming to feed. I just wanted to find the carcasses, see who's feeding on them or not. Yeah.
Thank you. Thank you for a great talk. I had difficulty keeping track of the colors and numbers, and it was quick. But so I was wondering, when there is a big carcass, it stay longer time each stage, and it goes to much more advanced stage of decomposition, because the small one, they cannot, because they will be eaten before they really decay. So how much is really because of the size of the carcass itself, versus being able to really get to stage of high decomposition. Like if you imagine a small one that would not be detected for a while, but then would start to really rot and all that, would it attract the same kind of predators, scavengers than the big one, in fact?
So what was interesting to me was actually like in terms of spreading in terms of carcass decomposition and also making the stages. There is no research that is stages like that. That was what we're trying to propose. I hope it gets accepted as a standard two-stage carcasses that decompose. The small carcasses, if they are lasting like 7 days in the field, I would consider that as stage one, because they don't entirely decompose to a part where you have the skin and bones only like remaining. After 7 days, if it's a small carcass, for example, like an antelope, even if no scavenger feeds on them, there's a high chance that a lot of protein or flesh is still there for any species to feed. And there's a high chance that even the generalist species that I put, they're still going to feed on it, because a lot of meat is still available and some fluids are still there. Whereas after 2 weeks, well, it won't last that long, but then after 2 weeks, that's when you start to see that the liquids are mostly like gone, they've evaporated, and it's just the skin and the bones. Oh.
Is there a question?
Thanks for your talk. Did you observe any change depending on the season, whether it's the wet season or the dry season, maybe?
Yeah, so also that is what I want to study for my PhD. Well, since now I'll have GPS collars, I'll probably find more carcasses. So the elephant carcasses that I followed were dying naturally. And most of them are affected by climate-related causes like heat, so which usually happens during the dry season. And during the wet season, there's a lot of food. Most of the adult elephants were not like dying a lot, and you won't find any carcasses during the wet season. I did drive around as well in the wet season, so most of the time I didn't find any carcasses. So I would assume that during the wet season, you won't find as much as the dry season. So, but also it's part of my PhD study to find out if there's a difference in carcass accumulation between the wet and the dry season.
Thank you very much. Thank you, Teddy, for your presentation. So it's time for coffee break. It will be a 15 minutes coffee break. Okay, so we now welcome Louise Lowe from Villefranche-sur-Mer Oceanography Laboratory, and she's going to present us the long-term monitoring of zooplanktonic communities in the Bay of Villefranche-sur-Mer in the context of climate change over 55 years.
Thank you. So, hello everyone, I'm Louise Lowe, and today I will present you one part of my project that is entitled Long-Term Monitoring of Zooplankton Communities in the Bay of Villefranche-sur-Mer in the context of climate change over 55 years. So global changes affect marine ecosystems through many ways, by impacting the temperature or the salinity, for example, and those environmental pressures affect marine diversity, especially plankton, that by definition defines all living organisms that cannot move against currents when facing unfavorable conditions. And that comprises phytoplankton, primary producers performing photosynthesis, and zooplankton, animals. So due to its high sensitivity to environmental pressures, plankton is said to be a good sentinel of ecosystem state, and moreover, it is at the basis of marine food webs, and it plays a major role in carbon cycle. So for those reasons, it is essential to monitor plankton nowadays in the context of global changes. To do so, long-term time series are a very useful tool as they enable to link interannual variations in plankton communities with environmental pressures, and several studies in the world have already shown interesting correlations between changes in the environment and changes in zooplankton biomass or abundances through time. So among the regions in the world that are very well studied in the context of global changes, there is the Mediterranean Sea, that is said to be a hotspot of global changes. And more precisely, in the Mediterranean Sea, there is the Point B station, located at the entrance of the Bay of Villefranche-sur-Mer. This station is influenced by the Ligurian current, characterized by a low chlorophyll A concentration. And the time series at the Point B station is said to be historical because it is one of the longest zooplanktonic time series in the world, with sampling starting in 1967. So previous studies in the past have already focused on one part of the time series, but none of them has studied it in its whole. So this is the goal of my project, to describe the series since its beginning until nowadays, in order to characterize long-term trends in the context of global changes and to provide a better understanding of ecosystem functioning. So to do so, I work with data that have been sampled at the Point B station. So I have environmental variables such as temperature, oxygen, that have been sampled using several devices deployed through the water column and measuring the different parameters at several depths. Then the environmental variables have been processed and treated. And then I also work with zooplankton samples that has been sampled bi-monthly since 1967, using a vertical net deployed in the water column and collecting plankton from 75 to 0 meters depth. Then the plankton is stored, and then thanks to the ZooScan that you can see here, it's a quantitative imagery device, it is possible to acquire images of all of my samples, and after a processing image step, I have access for all of my objects, so for all of the images in my samples, to features related to traits at the individual level. And moreover, the goal for me is also to study taxonomy. So for this, what I did is that I selected 3 dates per year for which the taxonomy group of each image, so of each plankton organism, has been validated manually by experts of the lab in order to train a deep learning model on those 3 dates. Then it enabled me to predict the taxonomical group of each of my plankton organisms of my whole time series. So I have environmental time series and I have plankton time series, so morphological ones and taxonomical ones. And then I could treat my time series the same way. And it enabled me to obtain several interesting results. So first, regarding the environmental long-term trends, here you have the deseasonalized component. And at 10 meters depth, I observed a significant increase in the temperature with more than 1.5 degrees in 55 years. So that's a huge increase. And I also observed a significant increase in the salinity. So following up on this, I had a look at the stratification index available since 1995. That is defined as the difference of density between the top and the bottom of the water column. And here again, I observed a significant increase in the stratification, indicating a water column that is less mixed through time. So I was wondering how this could affect the primary production, and I had a look at the chlorophyll A, that is a proxy for the phytoplankton, and I observed a significant negative trend in the chlorophyll A concentration. So I was wondering what inside my phytoplankton community could explain this. And thanks to the HPLC data that are available since 2012, I have access to the 3 different size classes of phytoplankton, so microphytoplankton, nanophytoplankton, and picophytoplankton. But as you can see, I did not observe monotonous significant trend for the 3 size classes of phytoplankton, but this can be due to the fact that this time series is much smaller than the one we have for the whole chlorophyll A. But after noticing those changes in the environment and primary production, I was wondering how this could affect the zooplankton. So I calculated the total zooplankton concentration per date, and I conducted a trend analysis on the whole time series that was not significant. So then I conducted a breaking point analysis to see if there were some dates indicative of significant changes in my time series, and 4 dates appeared as significant. So after further investigating on them, I noticed there was a significant decreasing trend from the breaking point of 1999, indicating a zooplankton concentration decrease over the last 20 years. So I was wondering what inside my zooplankton community could explain this. And I focused on 5 major groups that play a major role in the ecosystem functioning. So gelatinous filter feeders that are tunicates, gelatinous carnivores like jellyfish, chetognata that are top predators, Rhizaria like foraminifera, and copepods that are crustaceans very abundant. And as you can see from the breaking point of 1999, I did not observe significant monotonous trend in the 3 gelatinous groups, but I observed a significant increase in the Rhizaria concentration and a significant decrease in the copepods concentration. And the copepods are very abundant, and their dynamic is pretty much the same as the whole community, so it's quite consistent to observe the same signal. So we see that we have different trends depending on the taxon and functional group. But thanks to the ZooScan, I also have access to traits at the individual level, enabling to access intra-taxon variability. So then I decided to focus on the copepods because, as I said, they are the most abundant groups in my community. And I computed the mean area that is a proxy of the size of my copepods per day. And I conducted a trend analysis, again, that was significant. I had a significant increase in the mean area that was quite confusing in the context of an increase of temperature and more oligotrophic conditions. So then I decided to do a size spectrum analysis to see what was happening more in details. So it's an approach that is commonly used in marine systems, and that is based on the fact that we have an exponential decrease of abundance of plankton with body size. So this property is encapsulated in what we call the normalized size spectrum, enabling to calculate the abundance per increasing size classes. So I calculated my size spectrum for my copepods at each date, and then I was wondering how the slope of the size spectrum was varying through time. And I observed a significant increase of the slope, indicating a slope that is less negative, so a size spectrum that would be more flattened. And then I was wondering, what about the intercept? If the intercept would be the same, or if the intercept would be smaller, but with the slope more flattened. And what I observed was a significant decrease of the intercept, indicating that we have a more flattened size spectrum and that the smaller organisms would be less abundant. So to confirm this, by looking at what was happening inside my community of copepods, I classified my organisms depending on their size and trophic gills. And what you can see here is that I did not observe significant trends in the big herbivorous or big omnivorous in concentration, but I observed a significant decline in small herbivorous and small omnivorous concentration. So if the smaller ones are less abundant, then it's normal that the mean area of the community increases. So to summarize, with this work, we've seen that with climate change, we have increase of environmental pressures with changes in the temperature and the salinity that affects the stratification. And this has effect on phytoplankton, but also on zooplankton, either on the community composition with different trends depending on the taxon, but also on the size structure with a decrease of small copepods. And one hypothesis I have for this decline would be that we have an increase of predation pressure on small copepods. And to test this hypothesis, one perspective would be to use convergent cross-mapping. That is a statistical tool enabling to look for causation links between variables and not only correlation links. and that would help me to see if the potential predators of those small size classes are causally linked with the decline of the small copepods. So thank you for your attention.
Thank you for your talk. So I didn't.
I think it will impact the whole structure of the community, because in the plankton system, we have the potential, like the size that is a major trait and that defines the potential prey that you can have. So based on your size, you won't feed on the same prey. So if we have a decline of the small copepods, either it means the predation pressure is higher on them, or either it might be a bottom-up effect, so less phytoplankton that are eaten by those small copepods, and so then less small copepods, and this can affect the whole trophic structure ahead.
So for the last result, you said maybe there's increase of predation pressure on small copepods. I barely remember something like the cyanobacteria being too big to eat for some zooplanktons. And I assume that cyanobacteria is also increasing due to temperature. Do you think it might also be that like maybe the mouth opening of the copepods is not enough to eat the big phytoplanktons, and this could also be like a bottom-up effect rather than top-down?
That's an interesting question. So just to come back on this, so as I said, I did not observe significant trend for the size classes and the cyanobacteria. They are in the phytoplankton, but in other sites in the Mediterranean Sea, they observed a significant increase of the phytoplankton, so increase of cyanobacteria, and decrease of microphytoplankton, so diatoms. And it's rather the diatoms that are the preferred food for copepods. So I would say, like, if diatoms are declining, then the copepods that eat diatoms might suffer from this bottom-up effect. They will have less resources to feed on. But here, with what I have, I don't see it.
You looked at the salinity and temperature. I was wondering if the dissolved organic carbon might also be changing over the years, or is there anyone looking at that, or would that maybe affect also these communities?
Yeah, I did not have the dissolved organic carbon in my data, but I observed a decline of the particulate organic carbon. But yeah, this might also suffer from this, of course. Thank you.
Thank you, Luis, for your presentation. We are now welcoming Lea Kering, affiliated with Caen University and working at the research unit ID. And she's going to present us the shifts in the condition for pollen diffusion related to fruiting dynamics in temperate oak species as a special regional and temporal perspective since 1960.
Thank you for the presentation. As you said, I'm currently working at the ID lab in Caen, but today I'm going to present you the work I did during my PhD in the biometry and evolutionary biology lab in Lyon. So first of all, you have to know that temperate oaks exhibit masting. Masting is a reproductive strategy which is widespread among tree species that consists in a fruit production that is highly variable at the individual scale and highly synchronized between individual. Here on the graph, you can see the fruit production over the years of 10 trees of the same population. As you can see, there is a high interannual variation in the amount of fruit produced, and the trees are fairly synchronized. Fruits constitute a pulsed resource which has cascading effects with ecological consequences on the consumer's population dynamics and also consequences on economy and public health. It is therefore of high importance to understand how masting works. So if we take a closer look at this graph, first focusing on these high production years, we can see that they are systematically followed by low production years, which are a direct consequence of the previous year high production. But we can see that these low production years can sometimes be followed by other low production years. And in this case, the low production is understood as a fruit production failure. These fruit production failure introduce a level of irregularity in the fruiting dynamics. And this irregularity is key because it is the component that allows for controlling the consumer's population and increasing the reproductive success of temperate oaks. So if we want to understand what is driving the fruiting dynamics of temperate oaks, it is of high importance to actually understand what leads to these fruiting failures. In temperate oaks, the fruiting quantity is determined by the amount of pollen that is available for reproduction because they are wind-pollinated species through a phenomenon called pollen limitation. And it has been described that the major case for leading to fruiting failures is pollen diffusion that occurs in weather conditions that are unfavorable. Typically, it rains, so the pollen is stuck on the ground. What determines these conditions for pollen diffusion is the timing at which pollination occurs. In my work, I refer to this timing as pollen phenology. And the current pollen phenology leads to pollination occurring in weather conditions that are frequently unfavorable for pollination, which leads to irregular dynamics. So, if we want to understand what fruiting dynamics are going to become in the context of climate change and what they are becoming in the current of already ongoing climate change, we can imagine two extreme scenarios. The first one is that pollen phenology would be unchanged and pollination conditions could become more and more favorable for pollen diffusion, leading to less and less frequent fruiting failures and thus more regular fruiting dynamics, which would be a bad thing for temperate oaks. The other extreme scenario we can imagine is that phenology would become earlier because it is itself driven by weather conditions, which could maintain a variability in pollen diffusion conditions that is similar to what we know today and maintain some sort of irregularity in the fruiting patterns. So in my work, I tried to understand what has already happened in the context of recent climate change. And to do that, I worked in two main steps. The first one was to describe the whole chain of consequence I tried to describe you in my introduction, from pollen phenology to pollen limitation. And then I tried to retrace the trajectory of this whole chain of consequence since 1960 through extrapolation of the whole chain. To do that, I had access to a pollen data set that was given to me by the French Network for Aerial Surveillance, which gave me access to daily pollen, the daily airbound. The daily amount of airborne pollen for 48 cities through the whole France. So this is an example of what it looks for 1 year for one city. On this pollen distribution, I computed pollen phenology, which was the beginning of the pollenic wave. And I also computed pollen concentration, which I took as a proxy of pollen limitation. I also had access to a forest cover database and also a weather database, but I'm not going into much details here. So if I go back to my initial goals, first I'm going to describe how pollen limitation is driven through data analysis, which is going to be 3 steps. The first step was to develop a model that allowed me to predict pollen phenology. I'm going to go very quickly through this. It was a whole work, but basically I used a tool, which is the pollen phenology, the phenology modeling platform developed here at the Ceph. It is developed to predict leaf out phenology, and I adapted it to predict pollen phenology. But basically, all you have to know is that in order to predict pollen phenology, you have to use the temperature at the end of the winter and the beginning of spring. So now that I know pollen phenology, I know when pollen diffusion occurs, and I can identify the pollen diffusion period. To do that, I used a sliding time window analysis, which means that I computed the temperature on several time windows and found the one that best correlated with the pollen diffusion, so the various ones. And the best one started 19 days before pollen phenology and lasted for 30 days. Okay, so now that I know when pollen diffusion occurs, I wanted to describe how pollen limitation respond to the conditions at this timing of pollen diffusion. And so it was this graph. I looked at how pollen concentration responds to the diffusion temperature and tried to find the function that best describes this response. So this is the function. And I took this function, I called this function pollen diffusion coefficient. Okay, so now that I know how pollen phenology determines diffusion conditions and how diffusion conditions determine pollen limitation, I led a 60-year simulation to see how pollen limitation evolved. So if you got lost, it's the time you can come back. So in order to assess pollen phenology, I just needed weather at the end of winter and the beginning of spring. To know diffusion conditions, I only needed the weather during the pollen diffusion period. And which I had for all these points, which were the cells, the Météo France cells for which we have weather, in which we also have temperate oaks. I didn't do the work for the other cells because they did not interest me. Yeah, and once I know the diffusion conditions, I can use my pollen diffusion coefficient function to assess the pollen limitation. So when I look at the result, we can see that we predict that pollen phenology has shifted since 1960, becoming earlier and earlier, which is what we expected, and that it became earlier for all France. Despite this pollen phenology becoming earlier, we had an increase in the pollen diffusion conditions, with weather conditions becoming more and more favorable for pollen diffusion. So pollen phenology did not advance enough to maintain stable diffusion conditions. Okay, but the cool result is what I told you at the very beginning of my speech, is that what counts for masting when it comes to masting is that it has to be variable between years. So we looked at the variation between years in these diffusion conditions, and we saw that they have shifted very differently between France. With in the north of France, they became more and more stable and more and more favorable. No, sorry, they became more and more variable, and at the south, they became more and more stable, which was cool because we expect that masting will respond differently in these two areas of France. And what was really, really cool is that in another independent study on another data set, a colleague of mine showed that fruiting dynamics are actually different currently in France between the northern side and the southern side, with northern side having more irregular dynamics, which is consistent with diffusion being variable between years, and the south of France, the fruit production dynamics are more regular. So what my work shows is that this pattern have probably appeared quite recently, and that so it shows that when you work with masting, you have to take into account that what you see today is probably already the result of past climate change. Thank you for listening to me.
I really find your study very interesting. I have a simple basic question. That machine that records like the pollen passing through, like does it also differentiate the different pollen like from different plant species, or also like is there like speed which pollen moves with the wind? I don't know, like new to me. Or maybe can you please just describe the machine and how it works, how you collect pollen?
Okay, so the machine is called the Hearst capture, and the way it works is that it is not it is directed in front of the wind, and there is some sort of box at the beginning of it, which standardized the speed at which air comes into. So these are comparable to each other despite wind speed that varies between days. Does that answer your question? Okay. And on the part about different plants, you asked me about that. So there are technicians who come to the machine and take the pollen, and then they identify the pollen at the lab. So that's how we know which pollen is from which species. But when it comes to oaks, we can only go to the genus. Yeah, for all year.
Thank you for the talk. It's really cool actually what you have been able to demonstrate with this pollen and the oak system. I like it because I work with mice, so they actually depends a lot on the hogs. I was wondering, did you try by curiosity to do prediction? Because I guess you can get some pretty good prediction of like the future weather that will happen in France with the current modeling. So could you did you play a bit with like doing some prediction or not yet?
Not yet, because this work happened like maybe 3 months before I turned my PhD thesis. So it was quite a surprise how cool it was. And but the plan is to do it in the future. But as I'm already doing a postdoc on the side, it's a bit too much for now. But it's going to happen, I hope.
Okay, nice. We'll be looking for this. Thanks.
The question? Thank you very much for the talk. I was wondering, you said that most of the fruit production is driven by pollen limitation or not. Is there also a component of resource limitation? And if so, is it likely to become more and more important as the climate changes?
It is heavily debated. There is evidence that for common beech, it's the case. For oak, the amount of resource available definitely plays a role, but it is debated whether it is a cue or it is a physiological impossibility to invest. So when at the beginning, I did not go into too much details because you can always go into more details. But when I talked about the years where there is a low pollen production, sorry, I forgot the word. The arrows showing the low production years happening right after the high production year. Here, the low production happens because of the low resource availability for the tree. And so it is debated if it's a cue or if it's a thing. But the thing is that there is no sign that it is heavily costly to produce fruits. So I don't think that in oaks, I mean, and so I don't think that in oaks, the increasing of maybe resource limitation will be a thing in the near future.
Thank you very much.
Thank you.
Thank you, Leah, for your presentation. We now welcome Camille Touchet, affiliated with Claude Bernard University, Lyon 1, and working at the Laboratory of Ecology of natural and anthropized hydrosystem. And she's going to present us the impact of the plastic-associated biofilms on primary consumer Fith acuta. Is that the right pronunciation?
Yes. So hello everyone. I will set my time so I'm not too long. So I'm Camille Touchet. Thank you for the introduction. And today I'm going to present you part of my PhD work that I did at the Lena Lab in Lyon on the impact of plastic-associated biofilms on the primary consumer Fith acuta. So to begin with, plastic is considered a material indicator of the Anthropocene, and its production is currently more than 400 million tons per year, and on all plastics we ever produced since the 1950s, only 9% have been recycled and 12% incinerated, meaning that nearly 80% of plastic accumulates in the environment where it's known to spread and fragment and spread to all ecosystems. And one of the major issues associated with this material is the wide diversity of chemical composition due to the mixing of the polymer resins with chemical additives to modify their technical properties. So these plastics in the environment are new substrate for microbial communities to colonize them and form the plastisphere, which is a plastic-associated biofilm. It's a community of microorganisms like bacteria and algae that live together in a matrix in addition to a surface, in this case plastic. This plastisphere, so this specific biofilm, has a distinct microbial composition and functioning. Some previous studies showed that there is a reduction of bacterial and algal diversity in these biofilms, and other parts of my PhD work showed an increase of the esterase activity and a decrease of the beta-glucosidase activity of these biofilms, so metabolic differences too. Freshwater biofilms in general, they play a central role in nutrient cycling because they host primary producers, they support the trophic network, and also they participate in the biogeochemical fluxes of carbon and nitrogen, and thus to the organic matter recycling. And so my work question was, does this plastisphere affect biofilm quality and consumers? Trophic networks, they regulate energy flows and ecosystem functioning. And the relationship between biofilm and primary consumer is a key relationship influencing ecosystem functioning. So the biofilms, they can influence their consumers, so the grazers, growth, abundance, and diversity, as they are a food source that can change in quantity and quality. And the grazers, they can also influence the biofilms while grazing. They can change the microbial biomasses and composition. So to answer my research question, I conducted an integrative study of the impact of plastic-associated biofilms on primary consumer Fith acuta, which is a freshwater snail that you can see here and on the picture. And the first thing I studied was the biofilm quality. I had the hypothesis that the quality of this biofilm would be lower than plastic with a reduction of nitrogen content, which is limited in freshwater. I also studied the snail phenotype and fitness, so I was thinking that due to the reduction of the quality, the snails would have to consume more biofilm, and I was also expecting their growth and fitness to be negatively affected. And I also look at microplastic exposure. I was thinking that snails would be exposed to microplastic through grazing, and therefore that I would recover the plastics particle in their feces. So to answer, to test my hypothesis, I started by doing a field incubation of biofilms. So I took 4 plastic materials that represent the 4 more produced plastic polymers. So we have polyethylene, polypropylene, polystyrene, and polyvinyl chloride. And I also took a control material, which is frosted glass. And all of these materials had a standardized surface roughness to avoid the physical bias. So here you have a few pictures of the process. So you can see my incubation devices. They were floating, and you can see that after 6 to 8 weeks, the plates are fully colonized with biofilm. So we analyzed this biofilm to do the elemental analysis of carbon and nitrogen. Then we conducted a feeding experiment during 3 weeks where we fed 20 snail individuals per material with the biofilms, and we renewed the biofilm every week. So here you have a few pictures also of the experiment and of one of the snails. And we did weekly individual measurements. So the first thing we measure was the biofilm consumption. It's a bit tricky, but you will understand. I took pictures before and after a week of grazing, so you can see that the plate becomes lighter after a week of grazing. And I worked on the color of the colonized plate. I took the RGB value and I converted it in relative luminance using this equation. And this relative luminance is a good estimate of biofilm thickness. Therefore, the before-after difference give me the biofilm consumption. So the more there is the difference, the more it's consumed. I also looked at snail phenotype and fitness. So I measured the body mass and shell growth every week, and also the feces production that I will not have the time to show you today. After the experiment, I allowed the snail to mate, and I measured some fitness parameters like eggs, clutches, and offspring produced. And I also extracted the microplastics from their feces, and I characterized the polymer of this microplastic using FTIR analysis. Obviously, I did some statistical analysis of all of this data, and now I'm going to show you a bit of the results. So the first thing I want to show you is regarding the biofilm quality. So we didn't have a real global plastic versus control difference for this parameter. Only the polyethylene had a lower nitrogen content. So here I'm going to show you the figure. So you have the nitrogen percent per mass of biofilm, and you can see that the control is on the left in beige and the plastics are on the right in black. The black box represents the plastic that is different to the control significatively. So you can see that only PE was different. And we didn't have an effect on the carbon content. Then, on the consumption, we observed a decrease of the consumption of all snails feeding on plastic at day 21, so after 3 weeks of experiment. So here you have the difference of relative luminance, so keep in mind that the more difference, the more consumption. And you can see that on day 21, all groups of snails feeding on plastic were consuming less biofilm than the control. Regarding the growth, we looked at the body mass, and we also had an effect on 2 groups of snails feeding on plastic at day 21. And on the shell length, we also had an effect on 2 groups, but it wasn't the same groups, and this effect was constant through all the experiments. Regarding the fitness parameters, we didn't have an effect. So for the record, I'm showing you the egg production, so you can see that there is no real difference between the control and the plastics. Regarding microplastics, I was able to show that snails feeding on those plastic-associated biofilms were grazing microplastics. So it's a brand new result that I just got. You have the proportion of each polymer recovered in the snail's feces, and you can see that for each plastic group, like PE, you have 53%, I don't know if I can show it, no, 53% of PE was recovered in snail feeding on PE, and same for PP, PS, and PVC. For all those snails feeding on plastics, we recovered the plate's polymer as the main polymer in their feces. So these snails are actually ingesting microplastic through grazing. So to summarize these results, regarding the biofilm quality, we didn't see a strong difference on plastics compared to control. But we saw that snails feeding on the plastisphere were consuming less biofilm and were also growing less, but their fitness was not affected. And we saw that snails were exposed to microplastic through grazing and that we could recover the plate's polymer mainly in their feces. To conclude on this experiment, snails feeding on the plastisphere consume less biofilm and graze microplastics. Their growth is affected, but the snails show different responses among materials, and their fitness is not affected. So it leads me to a new question. Is there a difference in their energy allocation strategy that could also affect the population dynamics and the whole trophic network? So this is to investigate. So I would like to thank all my supervisors and collaborators, and I would like to thank you for your attention.
Thank you for the presentation. I have a question about like the biofilm that develop on the plastic tube. Did you check at the beginning of the consumption experiment if all the tubes had the same amount of algae growing on the tube? Because I would assume that maybe in function of the plastic tube could have a difference of how much biofilm you have, and that could also make differences in how much the snail feed on this specific tube.
Yes, in fact, we checked this also with the color, and we had the same colonization on all the plates at the beginning of the experiment.
Okay, cool. Thanks.
So I was wondering, so they have microplastic in their system now. How do you think this affects them in the way that they couldn't grow anymore? Do you think they didn't grow anymore just because they stopped feeding earlier, or is the microplastic somehow intervenes with some system inside the snail?
It could be a mixed effect, because we saw that they were feeding less, but we saw only this effect after 3 weeks. So maybe we start to see this effect when they are big enough to eat a lot, so we can see the difference. But also the microplastic, they could be stuck in their digestive system. It doesn't seem to be the case regarding the feces effect, but there could also be a chemical effect of their composition that we don't really know. So it could be a mix of all these effects that could explain why they react so differently.
Thank you for the talk. I have some questions about the mating protocol. You said that you mate the snails with an albino mate? Yeah, and did they came from the same sample, exposed to the same plastics or no?
No, the albino partner wasn't exposed to plastic. It was reared as a routine in the lab. In fact, we used the albino partner just to see after if the babies are pigmented or not, to see which snail function worked. So in this, because they are hermaphrodites, so they can be both female and male. And the plan at first was to assess the female part, and the albinos was kind of a control. If all the eggs we had were albino, it would have meant that the snail didn't reproduce, in fact, because they can also self-reproduce.
Okay, thank you. No more question? Okay, thank you very much. Thank you, Camille, for your talk. We now welcome Julie Sénécal, affiliated with the Biology Center for Population Management and Development Research Institute. Okay. Oh. And she's going to present us the contribution of gut microbial communities to insecticide resistance in Spodoptera frugiperda. Is that right?
So hello everyone, I'm Julie Sénécal. I'm in my first year of PhD between two labs here in Montpellier, the CBGP and the GMI lab. And today I will talk about the contribution of gut microbial communities to insecticide resistance in Spodoptera frugiperda. So as we all know, invasive species are a growing threat to aviculture. A species is considered invasive when it was moved beyond its native range, has established a self-sustaining population, and can spread in the new environment, causing ecological or economical damage. And a large proportion of invasive insect species are agricultural pests. And according to FAO, the use of pesticide worldwide has doubled between 1990 and 2022. But the widespread use of insecticide has accelerated the development of resistance in pest insect population. And insects have a different mechanism of resistance. It can be a behavioral resistance, cuticular, metabolic, or even on target site. But today I want to talk more about the microbial resistance, which refers to the ability of the microbiota to directly degrade the insecticide or indirectly enhance resistance by modulating the host detoxification gene expression. And to study this mechanism, my study model is the fall armyworm, also known as Podoptera frugiperda, and in French, la chenille légionnaire d'automne. So the female will lay eggs on the plants, and when the eggs hatch, the larva goes through 6 stages before going into the soil to pupate. This pest comes from America. It has been firstly recorded in Africa in 2016, and 2 years later in Asia and then in Oceania. More recently, a few specimens have been found in the south of Europe, and this is a polyphagous pest that can feed on up to 300 host plants, but one of its favorite meal is the maize plants. So the main question of my PhD, how do gut microbial communities contribute to insecticide resistance in this species? But firstly, we want to know what the microbiome of Lepidopterian look like. And some studies said that Lepidopterian harbor a transient microbiota which change with the environment or the host plants, while other studies said that they have a resident microbiota with core member. And to understand which one is true in Spodoptera frugiperda is really interesting to understand how these bacteria will have an effect on insecticide resistance. And it's even more interesting when we know that worldwide this species is resistant to 21 insecticide of 10 classes. So the main objective is, the first objective is to assess the variation in insecticide resistance and gut microbiota composition across natural population. And today I will talk about the first step of my work, which is to assess if insecticide resistance level in fall armyworm vary significantly among the population and also across different classes of insecticide. This will be like the basis of my research. So later on I will link this data with microbiota. So today I'm not really going to talk about microbiota. So to do this work, last spring I went to Kenya to sample 10 sites. They were all irrigated maize field, and all sites were located in an agricultural area except for the site of Nairobi. We collected the eggs or the larvae directly on the plant. And back in the lab, I ran some essay on pesticide. I used two synthetic pesticides commonly used in Kenya against fall armyworm. These are emamectin benzoate and lambda-cyhalothrin. I use them at 3 concentration based on the manufacturer recommendation. So they are both neurotoxic. One activates the chloride channels when the other one blocks the sodium channels. I also added a biopesticide, which is rabbit urine, which is commonly used by Kenyan farmers when they can't use insecticide. And I also added a distilled water control. So in total, I have 8 treatments. So in the lab, I starved my larvae for 6 hours, and then I put a little piece of maize leaf and dip it in the treated solution. Then the larvae were fed for 24 hours on this treated leaf. For each treatment, I allocated 10 individuals for the survival monitoring and 10 individuals for the microbiota monitoring. I repeated this experiment 3 times for each population, and I sampled 10 populations, so I did that 10 times. Now for the result. Here we have the variation in resistance to different insecticide. When we look at the survival probability, we can see a lower survival probability under emamectin benzoate treatment, here in red, compared to the lambda-cyhalothrin treatment in green. And for the lower concentration of lambda-cyhalothrin or the rabbit urine, we have no difference with the control. So we do have a variation in resistance between this insecticide. And now we are looking at the percentage of individuals reaching each stage. And we can see that under rabiturin, lambda-cyhalothrin, and control treatment, we have more than 50% of the individual reaching the adult stage, with less than 25% under emamectin benzoate treatment, and up to 0% for certain population. So even if we have the same patterns of resistance for the dense population, we do have some variation between them that need to be analyzed closely. And now we are looking at the resistance patterns across population. We were hoping to see one of two patterns, either a trade-off mechanism, which refers that a trade-off is when a population is resistant to one insecticide, but this resistance has a cost, so it's lead to a lower survival to the other one. So in this case, we would have like a negative slope. Also, I was expecting, sorry, a high survival to the two insecticide. In this case, we would have all the dots right here, but we don't have any of these patterns here, and we have like a very heterogeneous profile across all the population, which means that the resistance mechanisms to these two insecticides are really distinct. So far we focused on the survival data, but here I want to focus on the sublethal effect, because sometimes the insecticide won't kill directly the insect, but it will affect the development of the insect. And here we have the larval development time between all the treatments. So we don't have any data for the emamectin benzoate higher concentration because all the individuals were dead. But we have, for the second one, we have a longer development time. And now when we look at the pupal mass, we have a lower pupal mass. This suggests a sublethal effect and an altered development in emamectin benzoate treatment. So if we go back to my first hypothesis, we do have variation in resistance between insecticide with a high sensibility to emamectin benzoate and a high resistance observed under lambda-cyhalothrin. The rabiturin show no measurable effect, and we have the same pattern of resistance across all population, but with some variation. And for the sublethal effect, we have high sublethal effect under emamectin benzoate treatment and low to no effect under lambda-cyhalothrin and rabiturin treatment. This means that we have a complete resistance under lambda-cyhalothrin, either on survival and sublethal effect. So now the goal is to link this resistance data with metabarcoding data. Remember when I explained the experiment, I said that I allocated some individuals for the metabarcoding and microbiota monitoring. So now I will need to link, to analyze this microbiota, to link the composition and diversity of the gut microbiota, to see if they are associated with the resistance level between the population. And the goal after that is to experimentally validate the functional role of the bacteria by assessing how they can either directly degrade or induce the expression of detoxification gene in the insect. So that's the work for the next year. And before I finish, I would like to thank the entire ICP team who helped me either on the field or in the lab. And also thank you for your attention. Any question?
Thanks for your talk. So you said that there is various ways to resist to insecticides. The one using microbiota, do you know if it is frequent or if it's rare?
The studies about microbiota are really recent, But on other species, they have found that some bacteria will degrade directly the insecticide inside the gut, but more frequently the presence of bacteria will enhance the gene expression. But I don't know if it's really common, but it's being more and more studied, so maybe we will know soon.
Okay, thanks.
Thanks for the talk. So I was wondering related to your result. So what is the best strategy then to reduce this resistance to the pesticide in this population? Is it to use the pesticide which is highly impactful on which individual will survive, or to actually use the pesticide which select a bit less certain individual?
It's a difficult question because the farmer wants the best result. So they will use the one which works the best. But I think, but it's really personal, the best way is to reduce simply the use of pesticide and maybe find alternative to those pesticides. Because for the pesticides that they are resistant to, the lambda-cyhalothrin, it has been used for a longer time than the other one. So maybe the resistance is from far away. And for the second one, which work best, it will the larvae will become resistant in a few years.
I see. Thanks.
Just a curiosity. Do you focus on the microbiome because you think that this is what most likely explain the resistance, or you are more interested just to see if the microbiome is involved? Like, do you have any evidence that in this specific species the microbiome could be linked to resistance?
Yeah, well, there is some studies that show that it could have an effect on resistance, but we are not sure yet. So it's yeah, we are not sure, but we need to work on that because in other organisms it has been shown that microbiota has a real effect, but not yet in this species.
Okay, and do you know if any of the other mechanisms that are connected to resistance actually operate in this species?
There is a lot of studies on the, well, it's at the beginning. There is studies on the target site resistance and metabolic resistance. I haven't seen on behavioral, but yeah, there is some on the other mechanisms.
In this species you're studying?
Yeah. No, not in this study. Okay. Other studies.
Thanks.
No more question? Thank you.
Thank you, Julie, for your presentation. We now welcome the last presentation of that morning session. So we welcome Pierre Boucher, affiliated with the Center for Research on Biodiversity and the Environment in Toulouse, with his presentation entitled Humans Use Morphologically Unique Freshwater Fish Species and Amplify the Risk of Functional Erosion.
So hi everybody, I'm Pierre Boucher and I'm a first year PhD student. And so yes, today I'm going to talk about freshwater fish species, but the important message is that I'm especially talking about human utilization of biodiversity and the consequences on functional diversity and functional structure. So first of all, human use biodiversity and used species share actually some particular traits such as high body mass or conspicuous appearance or high nutritional values. Yeah. And there is a very well-established literature about the functional consequences of extinction of threatened vertebrates or the functional impact of some human activities, but there is a lack of connaissance about the consequences of each human use on functional diversity. So we use the trait-based approach to trying, to fix this lack of connaissance. So first of all, before starting, I need to explain you a very important concept for the rest of the talk. It's called the morphospace. So here we have a multidimensional space where each axis represents functional traits. And I call this a morphospace because we use morphological traits. So here I present 4 traits like body mass, eye size, elongation, or mass size. But the important thing is that species that are inside the morphospace are where they are because of their traits value, according to their own traits value. And so each dot represents species. So now that we have this in mind, the question regarding this is how human uses filter functional traits, diversity, across the major taxonomy groups. And so do humans use species like evenly inside the morphospace, or do we focus on the most distinctive ones? And are those most at risk of extinctions? So we use freshwater fish species as a model, and we use the fish-based database. And why freshwater fish species? Because they are heavily used for food with fisheries and aquaculture, but for also ornamentation uses with aquarium trade, or for recreational use with game fish. And the 3 main drivers of my presentation will be which regions of the functional space are targeted by different human uses, whether exploited species are functionally rare or redundant, and lastly, how the potential extinctions of threatened species and new species can lead to the erosion of some functional traits and after the erosion of some ecological strategies. So first of all, freshwater fish species, they nearly cover half of the vertebrates diversity with 18,000 species. They contribute to key ecosystem processes like nutrient cycling or trophic regulation. And they are very essential to human utilization, ensuring food security, ornamental and recreational use. We use FishMorph database, and FishMorph database contain a lot of information regarding fish, and we use only morphological traits. And so here is the morphospace of all freshwater fish species. So you can see that there is here the big ones, here the small ones, and some weirdo over there inside the distinct region. So there is a very high diversity of morphologies associated to freshwater fish species. And what we need to know if the used species have occupied the same area in the morphospace. So actually we use nearly a quarter of all freshwater fish species on planet, and they represent nearly half of the whole morphospace. So here, it's the morphological space of all freshwater fish species that we use. And now for each different use. So we see that there is strong differences between the different use. For example, fisheries will target the entire morphospace, and game fish will focus on some specific area of the morphospace. And so yes, some uses span the morphospace and other occupy distinct region. So fisheries shares maximal morphological overlap with the other use. For example, 81% of the morphological space associated to aquaculture species are overlapping with fisheries morphospace. And in the opposite, there is some uses that shares minimal morphological overlap. So in other words, it means that there are some use that don't select any area of the morphospace, and other that will select very specific area of the morphospace. So now that I explained you a little this, I need you to explain another stuff, is the functional metrics that can quantifies how distinct you are from the rest of the species pool. So here in green,
you have like distinct species. And here is the rest of the species pool in red. And so we call this functional matrix the distinctiveness. And so the aim is to see like if human use the distinct species or if human use non-distinct species, commonly morphological species. So here are the results. We can see that first we will concentrate on the all uses. The more a species is morphologically distinct, the more it tends to be used. But it's not the same pattern from all uses. It's the case, and it's very strong for game fish and aquaculture that will select species with very high level of distinctiveness. And for fisheries and aquarium is actually not the case. Quite the opposite. So what we need to understand here is that unique species tend to be preferentially used by human, but human use select species with different level of morphological uniqueness. And now the last question is, so we use species and there are some species that we use and that are threatened. Are they like everywhere in the morphospace or are they concentrated in some area? So here what we see, if we remove species that are endangered and used, it's going to lead to functional erosion. So for example, if we remove critically endangered, vulnerable, and near-threatening species, we are going to lose like nearly 5% of functional diversity, and especially in this area. So the black area represents the area of the morphospace that we definitely lose. And we see that it's the case for the all uses, even if each use is associated to different region of the morphospace. So the global message is that we use biodiversity and especially freshwater fish for different uses. And human use disproportionately affects irreplaceable species and irreplaceable ecological strategies because we use morphologically distinct species. And this non-random selection focusing on morphological distinct species will increase the risk of losing unique ecological strategy. In other words, we impact directly the morphological space by removing irreplaceable functions. So if, for example, all the species that are threatened and used will disappear, we will lose unique ecological strategies that cannot be replaceable because there's no redundancy. So with that, what can we do? Being used by a human is not a fatality. It can provide some level of protection or conservation incentive. Why I explain it. If you are used by human need you because you provide essential services. So they might be more able to protect you. And we need to use this in conservation world. We need to have a trade-based approach as a conservation framework, and we need to prioritize conservation management on morphologically unique and functionally unique species. And we need to use human uses as a conservationist tool. You know, we use biodiversity, but we need to use this to protect biodiversity, like maintaining essential activity, because these activities that I show you are really, really important for human societies in an economical, social, ecological point of view. And we need to embrace the concept of nature for people by making these uses sustainable. So thank you for your attention. And before I finish, I really want to thank my two supervisors, Sébastien Brosse and Aurel Toussaint. And I also want to thank my friends and my colleagues that are here, Marius, Antoine, and especially Jade. Thank you.
Thank you so much. I was wondering, how are you able to disentangle like the human selection from the environmental filtering? Because we know that the environmental conditions are changing at the same time. So how are you able to?
No, we just take human uses without regarding any environmental filtering. It will be the next work. Here, the goal is to have like an overview of global trend all around the world. But the next step is to to see at a little scale, like biogeographic area, it's still huge, but to see if there is some correlation between phylogenetic diversity, functional diversity, and maybe if it's correlated to some environmental filtering. Of course, the human utilization of biodiversity is depending on some environment conditions. So yes, I think it's very important. Thank you.
Thanks for the talk. Really cool what you can show on this. And I was wondering, how do you categorize what each activities are catching, like fishing, recreational? How do you get this data exactly?
So the data come from FishBase. It's a huge database regarding fish, but it's a great question actually because it's a huge problem when we use this database because there are no metadata associated. So in the database, there are information about if the fish is used in fisheries, for example, if it's highly commercial or just subsistence fisheries. But for recreational fishing, for example, it's just a 0 or a 1. We don't know if it's like heavily used or just rarely used. So this is a big problem actually. And to correct this, we begin to do a web scraping, to have more precise information regarding these different uses. And in fact, I think we underestimate a lot the utilization of biodiversity. So the web scraping is not finished yet, but with the preliminary result, we see that maybe it's not 26% of the freshwater fish species that we use, but maybe much more.
Yeah, I was thinking about this, especially for the fisheries, because I guess they just declare what they officially are catching, but there is a lot of bycatch also with the many different fishing techniques. So yeah, I guess there is some good underestimation, but still cool patterns.
Thank you. And yeah, in fact, the Association of Fishers provide actual great tools to help understand how many fish that we took inside the river. But yeah, at an international level, it's super hard. For example, in North America, it's very well documented, but the game fishing in Southeast Asia is like we don't have any very precise information.
Hi Pierre, thank you very much for your talk. Very nice. Yeah, I was just wondering whether like to what extent the definition of your functional space is dependent on the traits you're using. Do you consider it quite comprehensive, or you think it will like improve with the database growing over the years?
That's actually a very good question because I did show you the first two axes of the morphological space. Here, for example. But for freshwater fish species, when we do the calculation, we need to keep four different axes. So I cannot show you here because at the end it's a lot of results. But for morphological traits, there is one key trait that shape the morphospace is the body mass. Like in every other organism, it is always a matter of who is bigger. But in fact, what an interesting thing to do is before we was like putting all the traits and doing the morphospace and functional space. And the problem is that because body mass is so important, we cannot see the differences between the other traits. So what they do now is if they take the trait space and they divide it by diet or life history, and we try to understand how these different aspects of functional diversity is more interesting or not.
No more questions? What? Yeah.
Hi there. I was just wondering if you looked at the geographical patterns of functional erosion, if different areas have a hotspot of loss of functional diversity.
No, not yet. And it's very, very important for two facts. Also because we don't use the same fish everywhere, and of course, and sometimes for the same species, we don't have exactly the same uses. And it's so different that, you know, for example, you take North America with game fishing and you take Southeast Asia for food security. It's the importance for conservation and management after is completely different. In a case, you can easily, you know, trying to regulate or control. On the other hand, you have population that completely depends on fishes to eat. And so at the level of both functional and human and social consequences is very important. That's why the next work is about defining a biogeographic area and measuring like functional diversity. So this kind of space, but everywhere in the world, and associated this with phylogenetic, taxonomic, no, not phylogenetically diversity. Yeah.
Awesome, great, thanks.
Okay, thank you, Pierre.
Thank you. I would like to thank all the presenters for the really interesting presentation and all the participants for the question and attention this morning. Just one information, you will find all the details